Hi everyone! So today we're going to be explaining how specialized carrier proteins in the inner mitochondrial membrane of brown fat cells allow those cells to oxidize fats to produce heat. A little background information. The production of ATP in mitochondria is dependent on the proton gradient set up by the electron transport chain. The flow of electrons is coupled to the energy-requiring process of pumping protons from the inner mitochondrial matrix to the intermembrane space. ATP synthase is a protein on the inner mitochondrial membrane that helps the protons flow down their gradient back into the mitochondrial matrix. This flow down the gradient produces the energy for ADP to become ATP. In addition to ATP synthase, there is another type of protein on the inner mitochondrial membrane in brown fat cells. The carrier proteins in the brown fat cells are uncoupling. These proteins do not have the enzymes to allow the protons down the gradient into the mitochondrial matrix. The gradient-busting chemicals then carry H+ across the mitochondrial membrane, which breaks the proton gradient formed during substrate oxidation. As a result of this, the organelle can no longer make ATP and produces heat. You can see this process in the first diagram, which shows how the uncoupling protein breaks the proton gradient and cannot convert ADP to ATP. This is why brown fat cells are known as biological heating pads. They are used to aid in the revival of hibernating animals and protect sensitive areas on newborn babies from the cold, such as the backs of their necks. In the second diagram, you can see a good representation of the ATP synthase being blocked in brown fat cells.